Welcome back, welcome back, welcome back to science. All right, guys, so in this section, it's not going to be as long as some of the other sections. I know you may be thinking, like, thank God. But ultimately, this is going to be about the cell cycle. The purpose of the cell cycle is just for growing. Once upon a time, you were one cell, and now here you are, 50 trillion cells later. Further, any kind of damage to any organ, any skin, any cell will be replaced with this process. The neighboring cell knows how to make that cell, and it will make that cell. It'll make an identical copy of itself, eventually fusing that gap, forming a complete continuous cell mat, if you want to think of it that way. Also, just replacing worn out cells. These are the focus of what's going on in the cell cycle. So, as a result of today's lesson, I want you to be able to tell me the stages of the cell cycle, what happens in each stage, also being able to identify, like, what do those stages look like, and lastly, like, why do we even care about the cell cycle? All right, guys, so that's going to be the focus on this chapter 3, section 4, the cell cycle. All right, so the cell cycle refers to the life of the cell. So the cell will live its entirety and create a second cell, and those are going to be identical copies of each other, and those are going to make other cells, and etc. And there's a series of events that are very predictable in the order. And I kind of think it's funny that we know so much about the different phases of mitosis. And really, it's drummed up to be this just like, oh, elaborate process. The reality is it's separating chromosomes, specifically these sister chromatids, to go to different places. And in the end, you're going to have identical cells. The reason we know so much about it is early scientists just couldn't get enough of this new tool, the microscope. And they were looking at cells all the time, and they were seeing them in these active phases. And so they looked and they could see the chromosomes in the mitotic phases, and they named each one of those like sub-steps within. And then there was like a long time where you couldn't see the DNA. They didn't know it was DNA, but the structures is what they were earlier looking at. And they called this like, well, this is the in-between phase, and they called it interphase. And the reality is that interphase, when a cell does like normal life stuff, it happens in interphase. Gene expression, largely interphase. So when the cell's routine, like why it exists, what it does, it's an interphase almost most of the time. So 90% of the time in the cell cycle, the cell is in interphase. All right, so what happens in interphase? Well, there's 3 different subphases there. Eventually, scientists like, something's going on here. And they eventually like, okay, I can see something is going on there. And as a result, I can see like nothing's happening, and then like the DNA's copied. We're going to call that the S phase. But before that, nothing's happening. We're just going to call that a gap phase, G1. And then after the DNA is copied, nothing happens. We're going to call that gap 2. They're wrong. But still, they stick with these names. And so interphase has 3 different subphases, G1, S, and G2. So let's take a look at those. So notice that we have interphase starting here on this wheel, moving across. And during interphase, you have G1, S, and G2. For a cell to go from one cell to 2 cells of the same size, it must first increase in size. Also, you think like, what about the things that are within the cell, the organelles? Those also have to be copied. What about the genetic material? That's the S phase copies the genetic material. And it stands for synthesis, which means to make. So in G1, the cell increases in size, and the organelles within it are copied. Most of them are. S phase, the DNA is replicated, so they're identical copies to each other. And in G2, final replicatory proteins are also copied. It's the shortest of these three phases, even though this diagram kind of makes it look otherwise. But you have replication proteins that are really getting involved, which include things like the centriole has copies of itself, and everything is gearing up, getting ready for this next phase. So 90% of the time of the cell cycle, it's interphase. From there, we get to mitosis. Mitosis has PMAT. The entirety can be remembered with the acronym IPMAT or IPMAT or IPMAT. However you want to remember it, it's that sequence. So let's go ahead and start talking about these mitotic phases and what happens there. Notice this. There's a checkpoint after G1. You're like, a checkpoint? What's that? To be continued. So in interphase, the cell is active. It's going to grow. It's going to copy its organelles. And it's going to get ready for the next stage. In the S phase, DNA is going to be replicated. And in G2, you're going to have the final things get ready for duplication. And now we get to the point of mitosis. So this is PMAT. During prophase, the genetic material that was an interphase was chromatin, but now it's going to tightly condense. And it's going to tightly condense around different kinds of proteins called histones and nucleosomes, and it's going to supercoil on itself and become really, really compact. And so something that was really, really thin, when you wad it into a really small area, that's visible. That represents the chromosome. Chromosomes condense in prophase. The nucleus dissolves. And so early microscopists were able to see cells in prophase and identify those chromosomes. And so that, like, hey, this is prophase. This is like, we're starting to see things here. Those chromosomes, which are actually sister chromatids. I'll show you. So those chromosomes, which are actually sister chromatids, you see the structure that's here. This is representing, so here we have a chromosome. It's going to be replicated. Now there's two. And whenever you see, you know how chromosomes kind of have that X-like look? It really is these two different structures here. These are called sister chromatids, is what they're called. They're genetically identical to each other, and they're going to be held together in this X-like look. During the process of mitosis, the whole thing there is to separate these, to put them in new cells, so the new cells are identical to each other, having the same original DNA, just copied. And then once it gets into new cells, the nucleus reforms around it, and the chromatid that's here is going to relax, be uncondensed, if you will, into the unraveled, readable, usable DNA. Okay, so for the structure here of the sister chromatid, they're going to line up, because there's 23 pairs of these, they're going to line up on the metaphase plate, which is an imaginary line in the middle of the cell. It's going to do this, so when they separate, you get a cell that has these, and then you're going to have a cell that's going to have these. And so you're going to end up with identical cells as a result of the process. The whole process of mitosis ensures that the genetic material is separated, and each cell is going to end with now two identical cells as the result of that one cell going through its process. So prophase, the genetic material goes from chromatin to chromosomes, so it condenses, it becomes visible for the first time. The nucleus dissolves. The centrioles are going to now have their spindle fibers connecting to this. In metaphase, they've lined up on this metaphase plate. So they've lined up in the middle of the cell. And the spindle fibers are going to start to pull this to the other side. And whenever it's starting to be pulled, it starts to move like this. And whenever you're looking at anaphase, it kind of has that A-like look. Turn sides, pull that stem out. And the reason it has the A-like look is if you have this, but you're pulling from here first, it's going to pull away with that kind of V-like look as it moves in that direction. So anaphase is pulling apart, and it has that visual look there. Further, once we get to telophase, the nucleus is going to reform around the new separated genetic material. The cell almost appears like a peanut in its kind of shape. It's not like a big circle. It elongated and started to pinch in the middle because eventually it's going to separate. But the genetic material is going to be within. It's going to reform in the nucleus. The genetic material is going to unravel back to chromatin form where it's going to be usable. And then the cell's going to go through a process called cytokinesis, which is the separation of those. You can see the word cytokinesis there. It's the division of the cytoplasm, and then we have two identical daughter cells. Here's an overview and a table form. And here is the image that I referred to. You can see the centrioles, you can see the nuclear envelope, you can see chromosomes that would make this prophase, you can see centrioles with spindle fibers, and you can see them late prophase starting to connect and push them in the middle. Now we're in the middle. You can see an anaphase, so that's metaphase. An anaphase, you're going to see spindle fibers moving the sister chromatids towards the centrioles on each side, and then you can have eventually the nucleus reforming, and then you have the chromosomes getting back to chromatin form where it can live life most of the time in that form. So cytokinesis is the separation of the cytoplasm. The name of that kind of pinching in is called a cleavage furrow. A cleavage furrow is this band, this ring of these actin filaments that are going to be around kind of tightening that, causing it to eventually separate. And you can see this elongation. Here's the cleavage furrow. Here's a deeper cleavage furrow on either side. And then eventually you have the cytoplasmic division, and you have the identical daughter cells. All right, guys, this is going to finish this subsection. I'll see you soon in the next. So long.